Okay, this is my first attempt at making one of these like summaries, like what we'd kind of go through in a biology workshop at the sixth form. So yeah, we're going to do DNA and protein synthesis. And it's me with Dr. Ellis. If you don't know my voice, apologies. Right, a good starting point for anything is the syllabus. So I'm going to address what's in the syllabus and no more. Well, kind of. It says the concept of the genome as a complete set of genes in a cell and of the proteome as a full range of proteins that a cell is able to produce. Those are just facts, so I'm not going to spend any more time on those whatsoever. Next is the structure of molecules of messenger RNA and tRNA. So I made a little diagram over here. Oops. I'm going to try and scroll. There we go. Right, so I've already drawn out mRNA and tRNA. They are polynucleotide molecules. So, you need to know like two similarities between them and two differences and structural as well. So in terms of how they are similar, I think, they are, well, the sugar that they contain is the same. Yeah, so they both contain ribose. Okay, and they both contain uracil as well, just in case it's a difference to DNA. Yeah, so RNA contains ribose and uracil. The differences is where they spend more time really. The mRNA, as you can see, it's a single-stranded molecule. However, tRNA is also a single strand. It's not like DNA where it's two separate strands. This is one strand, so I need to be careful with single-stranded. Okay, and it's the reason why this gets confused as single-stranded is because it is folded, and it's folded because it's got these red little dots here. Those are hydrogen bonds. So tRNA contains hydrogen bonds. So tRNA, but is folded using hydrogen bonds, but H bonds for short. So that's one difference. mRNA does not. I'm not going to write that as well. And the other key difference, again, a lot of people get tempted to talk about this. This is the anticodon, but it's not structure. It's just 3 exposed bases. Okay, you can see here I've shown all the bases as exposed. This would also have bases all over it, sticking out and sticking in, but these are the only important ones. That's the anticodon, but it's not structure. I'd always go for this, the fact that it can carry this blue box. So it must have something here, like a little hook, that enables it to hook onto the blue box. And the blue box is what tRNAs carry. It's an amino acid. So tRNAs have an amino acid binding site. So tRNAs has an amino acid binding site. Okay, so those are all similarities and differences between mRNA and tRNA. I've therefore fulfilled, let's try and scroll back to the syllabus, I've hopefully fulfilled that, the structure of molecules of mRNA and tRNA. Next is transcription as a production of mRNA from DNA, the role of RNA polymerase. Then it mentions about prokaryotes. I'm going to leave that for today, and I'm just going to look at the eukaryotes, about splicing. Basically, the prokaryotes is simpler, so we just have to make, if we learn eukaryotes, then we can just remove things for prokaryotes. And then translation is the production of polypeptides from the sequence, the role of ribosomes, tRNA, and ATP. And it says words like, and we just zoom through this. So I've got RNA polymerase to get into, mRNA nucleotides, transcription, mRNAs made, and pre-mRNAs there, splicing's there, translation, codons, mRNA, ribosome, tRNA, ATP. So I've got all those to address. Okay, so let's look at this diagram first. So this is showing and I have to think every single time about this, is it transcription or translation? So this is showing, oops, that's the wrong pen, that's my highlighter. So this is trans, Then I think I've got transcription and lation. Well, transcription comes first, and C comes before L in the alphabet. So transcription. This is transcription. This is DNA. You can see because it's double-stranded, totally. And then if I just label this, this is the enzyme. So this is RNA polymerase. It's in the syllabus. Make sure you know it. Okay. And we're in eukaryote, so this red molecule that it's making, this is pre-mRNA, and I'll go through that in a second. I'm just introducing, marking points. And names of enzymes get a mark. What the enzyme makes gets a mark. These look like lots of T's, but they are the building blocks of the red pre-mRNA. So these are RNA nucleotides. And you can see the RNA polymerase is only on one of the strands of DNA. It's called, again, you can kind of, you can call it the template or coding strands. Its proper name is the antisense strand. Okay, and I'll go through how to kind of link all those together in a minute or two. But I just want to show you how we can kind of like summarize all of this as one little diagram. So this is what I learned. If I'm making my revision notes before an exam, this is what I have, where a gene codes for a protein that controls a characteristic. This is the central dogma of molecular biology, the central belief. Okay, and I'm going to put a little thing on here. I'm going to put that on here. So this is my nuclear envelope. So out here is cytoplasm, and therefore in here is in the nucleus. Happy on that? Good, as you all scream good. So we've got our geography in our cell. And then the first thing that happens is, as we know, it's transcription. Where we are going to, I'm going to have to write up this slightly. So this is transcription that takes place in the nucleus. And I need to add things to it. I need to add the enzyme, RNA polymerase. And we need to add the RNA nucleotides. And we'll go back to that in a minute. I just want to give you an overview. And we make a molecule called pre-mRNA is made. But then leaving the nucleus, it's called mRNA. So we go from pre-mRNA to mRNA. And I go from precursor mRNA to messenger RNA by removing the non-coding bits. So we remove introns. Introns are removed. What do we join together? Exons are joined. Okay. So this here, this is the transcription bit. Curly brackets are always tricky. So that is transcription. Whereas pre-mRNA to mRNA, this is splicing. Some nice videos on splicing, but really it's 2 marks of detail is all you need. So for splicing, introns are removed from pre-mRNA and mRNA is formed. 2 marks. Transcription, we need to get 6 marks out of it ideally. Well, 6 marks is the biggest question we get asked in biology. And currently I've got 3. I've got RNA polymerase. If I add, moves along the antisense strand, that's 2 marks. And then I need to say about how RNA nucleotides are are attached, and I'll go through that in a bit. But crucially, pre-mRNA is made or mRNA is made. So making mRNA, how they attach, names of bonds, and then the name of the enzyme. So somehow I need to talk about hydrogen bonding and about phosphodiester bonds. So those are like the 2 other bits that you would add to this, but it wouldn't be suitable on this diagram. So that's transcription. I'm going to carry on and do translation, then go back to transcription to say how that happens. Because next is translation. So then this is translation. Where does it take place? In the cytoplasm. And what do we need to add? Well, I need to add charged tRNAs. So I'm going to put tRNA plus amino acid. Yeah, so the tRNA is carrying an amino acid. And we also need, well, I've already got mRNA coming in. I also need a ribosome. And I need to get 6 marks out of that as well. How do I get from mRNA with a tRNA and a ribosome to a protein? And that takes place in the cytoplasm. The protein then controls a characteristic. We're not bothered about that in this bit of the syllabus. We're just interested with transcription, translation. So finally on this, transcription, make sure that you know that it comes first. I do this little C there, and translation comes second. Again, they're easy to confuse. Okay, so let's just go back to that other diagram where we've kind of like labeled, let's see, isn't it? So if I was trying to get my 6 marks, and you can get them really quickly in biology. So I say RNA polymerase, I'll put pol full stop, 1 mark, moves along the antisense strand of DNA. 2 marks. 3 nucleotides, 3 RNA nucleotides. Well, they bind to the DNA. What bonds are they going to form? Well, they form complementary hydrogen bonds. 3 RNA nucleotides form H bonds with complementary bases on DNA. So I'm saying nucleotide there and bases. So that T shape is a nucleotide because it's a sugar phosphate base. But on the DNA, I've just got these sticky out bits, so there'd be a base on there, like a thymine. And while I'm on thymine, we can always get a mark for talk about what is the complementary base pairing. So if the DNA base is thymine, a RNA nucleotide with adenine will bind. Super. It's all probably worth mentioning uracil as well. So the base pairing, remember, if it's DNA, then if that's got an A base, then it'd be a U on RNA. So there's complementary base pairing. Yeah, remember there's no thymine in RNA. So we've done that. Then what I mentioned, the enzyme RNA polymerase. What bond does it form? It doesn't form hydrogen bonds. It forms the phosphodiester bonds. So the enzyme forms phosphodiester bonds. And then what's made? mRNA is produced. Again, I can say pre or just mRNA. So let's see if I've got the marks. I've got 1 mark there, 1 mark there, 1 mark there, 1 mark there. I probably gave the wrong example. I probably should have said base is adenine on the DNA, then it'll be uracil on the RNA. 1, 2, 3, 4, 5, 6. Again, I've never seen it more than 6 marks. It can be as little as 4 marks, but I've got them all. Crucially, learn your names of enzymes and learn your bonds. Hydrogen bonds, phosphodiester bonds. If I know the enzymes and the bonds, I've got 3 of the marks. And if you know what's produced, you've got 4 of the marks. 4 out of 6 as I reach for my calculator. I should be able to do 4 out of 6, surely. Yeah, 67%. That'd be a couple of marks behind an A star in an exam. So 4 out of 6 would easily get you an A in an A-level biology exam. Okay, so even if I don't know the antisense strand and I forget to write about the complementary base pairing or get that wrong, if I know my enzymes and my bonds and what's produced, then I'll get 4 of the 6 marks. I've therefore got a grade A. So that's transcription. Translation. Well, before we get to translation, let's have a look at translation. So this is a diagram that I got off the internet that shows, I've gone for the simplest one I could find. So the green thing is the ribosome. It mentions large and small subunits. Does it say about subunits in the syllabus? No, it doesn't. But these kind of latch on to the mRNA. You can see the code is shown, the dots saying it carries on. Don't have to worry about 5' and 3'. So if you want it, but it's never going to be an answer in a question. And this is our tRNA. And you can see the tRNA has got this AAG on it. So let's label that because it's not quite fully labeled. So this is our tRNA. This is a amino acid. And the AAG, this is the anticodon. And therefore the UUC, this is the codon. So the codon is a name for 3 bases, 3 coding bases on mRNA. The 3 important bases on tRNA is called an anticodon. The 3 in DNA is just called a triplet. So use the word codon for mRNA, anticodon for tRNA. And you can see, this bond here, this is a high energy bond. Oops, that's not it looks like a branch now. Sorry, I can't do it red. So this is a high energy bond formed using an energy molecule, using ATP. So that's the role of ATP, is to form that bond. Okay, so we need 6 marks for this. So let's have a go. So first thing, ribosome, 1 mark. Let's put binds to mRNA. Let's do it properly. So I've got 1 mark. Then I need to talk about this complementary base pairing. So a tRNA with an anticodon that is complementary to a mRNA codon binds. What bonds does it use to bind? Using hydrogen bonds. Okay, and then another tRNA comes in. And I can kind of say that, but I've no really need to, because this is just a cycle. What does the ribosome do to these? Because I've got multiple tRNAs come into the ribosome. What bond is this here? So let's get me red again for bonds. So this bond here, what's that bond that the ribosome is forming using the energy from ATP? Well, it's between amino acids. Yeah, the circles are amino acids. So that's a peptide bond. And that's a mark. Again, names of bonds get marks. So yeah, ribosome forms peptide bonds. Okay, polypeptide forms. That's a chain of amino acids, remember, until a stop codon is reached. So I've got 1 mark there, 2 marks here, 3 marks for hydrogen bonds, 4 marks for peptide bonds, 5 marks for it's making a polypeptide or protein until it reaches a stop codon. The other mark is, well, until it reaches. So the ribosome's moving. This ribosome is moving that way. Okay, So how far does it move? Again, I probably would have put it further before, but yeah, the ribosome moves one codon each time. That's why the code is described as non-overlapping, because if it doesn't move one base, it moves in threes. Okay, I always talk about, the fat cat sat. I read it in threes. If I read it in ones, I'd say the fa effa fat atka taka cat. Yeah, it would be nonsense. So yeah, that's why we have the yeah, that I'll get to in a minute, the non-overlapping code. Okay, so you can see here that AAG, this one here, this tRNA carries phenylalanine. They always have a three-letter abbreviation. So if I go to the codon map, oops, here we go. So it's fast scrolling on the screen for you. Sorry. This is a codon map. I know it's mRNA codons because it's got U's in it, and this is the first base of the codon, the second base of the codon, and the third base of the codon. Yep. So as it was before. So if I say AAA, well, that will call for the amino acid lysine. They are known by three letters, and you can see. So, for example, let's find one that's reasonably straightforward. Yeah, so serine. So AGC calls for serine. AGU calls for serine, and also UC, and then any other letter codes for serine. So there's 6 ways of coding for serine. That's why it's called a degenerate code. There's multiple ways for coding for the same amino acid. So Silvers also talks about experimental evidence for how we've worked this map out. So if I made some mRNA with the sequence AAAAA, well, it's read in threes. It's read in codons. So the codon is AAA. So AAA codes for lysine. So that would code for lysine, lysine, and lysine. So I could feed a string of A's into a cell and see what protein it makes, analyze that protein, and discover that, it's only made of the amino acid lysine, therefore AAA codes for lysine. So these are the early experiments that they did. Then they kind of went a bit crazy and did AC, AC, AC. So again, we split it into threes. And they found then that, ACA, well, that codes for ACA, so that's threonine. Again, none of these amino acids you have to know. And CAC codes for histidine. And then we're back to threonine again. And then it will go to histidine again, and so on and so on and so on. So again, they've worked out that it must be 3 bases that code for it, and they've worked out what combination of bases code for what amino acids by doing experiments by making mRNA artificially. I've been going a long time here. I wanted to try and stay under 20 minutes, but it is a big topic. But I'm almost there. So it doesn't say this here, but in another bit of the syllabus, it talks about that the code is universal, non-overlapping, and degenerate. And really, this is where the answers are to that. Universal just means that the same codons code for the same amino acids. So same codons code for same amino acids in all life. I put all in quotation marks just because it's technically not all. There's some very early bacteria that have a different coding system, but from E. coli bacteria to a giant redwood tree to a blue whale to me, we all have the same code. So I can put a gene from a spider into a plant, and the plant will make the spider's protein. It allows the science of genetic engineering, something we do in year 13. Non-overlapping is just the code is read in threes. So the code is read in threes, i.e. codons. So codon, codon. That's why I put it here. And why is the code degenerate? Because multiple codons code for the same amino acid. We've got 4 bases, and they are read as codons, therefore 3. So that's the number of combinations. I've got 64 different combinations of codons. Okay, so our picture here, there's 64 outer letters here, 64 of them. However, there's 20 amino acids. So there's 64 codons, and there's 20 amino acids. So we've got redundancy in the code, and that's the word for that is called degenerate. So just go back to the start just quickly to see what I've done and what I've not done, because I'm bound to have missed bits. Right, so give me a rubber. Does that rub out? It does. Oh no, it rubs it all out. I don't want that. Right, try again. That's better. Get rid of those things I've done. So I wanted to go through the structure of mRNA and tRNA. I think I've done that. Transcription is the production of mRNA from DNA. Yeah, there's 6 marks for getting that. The role of RNA polymerase makes phosphodester bonds in joining the mRNA nucleotides. Yeah, that form hydrogen bonds spontaneously because they have complementary base pairing. In eukaryotes, transcription results in the production of pre-mRNA. This is then spliced to form mRNA. I've done that. Translation is the production of polypeptides from the sequence of codons carried by mRNA, yes. The role of ribosome, yeah, it aligns everything together, and then it forms the peptide bonds. The role of tRNA, well, that carries the amino acid, and the role of ATP. Good. And so you should be able to relate the base sequence of nucleic acids to amino acid sequence of polypeptides when provided with a suitable data about the genetic code. I showed you that's that circly thing, but there's other variants of it. And interpret data from experimental work investigating the role of nucleic acids. I showed you that with those strings of A's or strings of alternating bases. Students will not be required to recall in written papers specific codons and amino acids for which they code. Wonderful. One last thing I just wanted to add. I know I've been going on for ages, but yeah, that tRNA is recycled. Yeah, so that will then, it goes, but then the tRNA will pick up another amino acid, and then it comes back again. So there's a little cycle there. Okay, I'll stop there because it's almost half an hour.